Good afternoon everyone. Myself Mrs. Ashwini Kadbhane from Department of Microbiology, SVKT College, Devdali Camp. Today we are going to see the TY BSc paper number 3, that is the Enzymology. In that we are going to see about the enzyme purification, and in enzyme purification we are going to see a method, that is the affinity chromatography, which is based on the specific binding method, that we are going to see in our today's session. So before moving forward, we are going to recap what we have already learned. We have already learned about the vitamins. We have seen their role, their occurrence, their structure and their biological functions. So before starting about the affinity chromatography, we have already learned about what is enzyme purification and why we need to purify the enzyme from the crude substrate. We have seen the different methods, and we have seen the enzyme solubilization techniques also. In our today's lecture, we are going to see the protein purification or the enzyme purification methods, which are based on the various criteria, that is the physical chemical properties of that enzyme. Okay, the purification of enzyme from the crude substrate depends on the enzyme activity also, and the basic chemical and physical functions. On that basis, a suitable method is used to purify the enzymes of our interest. Okay, in that we are going to see the principle and the method, which are based on their molecular size, on their charge basis, on the solubility differences basis, and on the specific binding property. In molecular size, we are going to see about the gel filtration method. Gel filtration, that is the molecular size. Various beads are used of the various size, that is the gradient, density gradient or the gel filtration. These both methods, they are used for the molecular size of the enzyme purification. In both method, gradient means according to the grade, according to the size, the grades, or according to the size the enzymes are to be purified, or according to the in gel filtration of gel is used. It may be a agarose gel or it may be a cephalophorus gel, and it is used on the basis of the molecular size to purify the protein. On the charge base, the ion exchange chromatography or the electrophoresis are used, in which the cation and anion exchanger are used to purify the protein on the basis of the charge. On the solubility differences, salting in and salting out method are used, in which the ammonium precipitate going to be formed. In salting in, the molecule is solubilized, and in salting out, the molecule is precipitate, and that precipitate molecule we are going to we are going to take that as the purified enzyme. Okay, and in that also precipitation method, solubility differences in that precipitation method is used. And last one, it is the specific binding property, such as the antigen antibody reaction or the enzyme substrate reaction, which is very specific. It requires a specific type of the molecule. Okay, so here in which specific binding, in this topic we are going to see about the affinity chromatography, in which we are going to use the various matrix, and we are going on that particular matrix. We are going to choose that matrix according to the enzyme of interest. The enzyme which we have to purify, according to that matrix is selected, and the enzyme is purified by using the affinity chromatography. We are going to see about the affinity, that is the principle and the working, its applications, its components, its advantages and the disadvantages. So here the learning objectives are the affinity chromatography, which is a specific binding type of a chromatography, such as the antibody antigen reaction or the enzyme substrate reaction. In such, we are going to use that for the enzyme purification, their principle, their working, their advantages and the disadvantages. So protein purification method, from protein or the enzyme purification method, from the crude we have to choose a particular method on the basis of the physical or the chemical properties of that particular protein or the enzymes, and from that we are going to purify our enzyme or protein of interest. It is the method, which is generally a multi-step process exfoliating a range of biophysical and the biochemical characteristics which depend on the solubility, charge, size, its hydrophobicity or hydrophilicity of the target protein. So here the protein purification method it has some characteristics. So it should be focused, that is high recovery rate. The protein which you have to purify, the recovery rate should be high because you are using the techniques for that. The reproducibility, the method which you are going to use, it should be reproducible. Again and again you can use the method because your sample would be more, the crude extract would be more. In one go or in one session you can't recover your protein. So it should be a reproducible method. It should be economical because you are using some chemicals or the reagents, they should be economical, and it should require a shorter period of time. Because if you are using a method and it takes a longer time, then it would be a very high cost method to purify the enzymes or the proteins. So it should be have a shorter period of time. In our enzyme purification method, already we have seen that the method is based on the various aspects or the physical chemical behavior of that enzymes. The aspects would be the molecular size, the charge, the solubility differences, or the specific binding property. Okay. On the specific binding property, the type of method which we are going to learn in this session is the affinity chromatography. Bioaffinity or the affinity. Bioaffinity is said because we are going to purify the biological molecules, that is the enzyme or the proteins. So it is called as the bioaffinity chromatography. It is one of the most powerful procedure in the protein purification. This method is highly selective because in this method we are going to use a specific type of a molecule to purify our target protein or enzyme. If we, for example, antigen and antibody reaction, a particular antigen have a particular type of antibody to kill. If an antigen is diagnosed in your body, a specific type of antibodies, they are prepared, they are produced by the B cells, and that antibody only can destroy the antigen. Here also we have to choose a specific molecule for your target or purify enzymes. So this method is highly selective as it is very specific. It is reversible interaction because you have to reverse the reaction to get your enzyme or get your protein. When there is a binding of a enzyme with a particular type of substrate which you have used, the molecule should be reversible. The reaction should be reversible because you have to take off your enzyme from that elute and you have to purify that. So it should be a reversible interaction. A classic affinity chromatography, it mainly focuses on the various interaction enzymes with the ligands. Here we are going to use the ligands to bind with the enzyme of your choice for the purification method. The ligands, it may be a substrate, it may be a coenzyme, it may be an inhibitor, or it may be an activator. Any type of molecules can be used as a ligands in the matrix for the isolation of the enzyme or for the purification of the enzyme. Now, before going towards the procedure, we are going to see what actually is affinity chromatography. Affinity chromatography, it is a liquid type of chromatography which is performed in a column. Okay, it's also called as the column chromatography because it is performed in a column in which matrix is poured and in which a ligand is poured for a specific type of reaction. Okay, affinity, it is a type of liquid chromatography for the separation, purification of specific analysis of a sample components. It utilizes the reversible biological interaction or the molecular recognition called affinity, which refers to the attraction. Affinity means attraction towards a particular molecule. Okay. So, which refers to the attracting force exerted in different degrees between the atom which cause them to remain in the combination. Now, here we have a solution which is having a combination of the molecules. From that combination of molecules, we have to isolate or we have to purify a protein or an enzyme of our interest. For that, we are going to use a molecule which has the affinity towards that target enzyme. So for that, we are going to use a ligand. It may be any, a cofactor, a substrate, a inhibitor, a activator, any molecule can be used which is very specific for our target, and by using that we are going to purify our proteins of interest. It may be a inhibitor, a antigen for an antibody, or a cofactor, or any. This method, it was discovered by Pedro Cuadras and Mir Wilczek. These two scientists have discovered the method, that is the affinity chromatography. The principle here also, as performed in your paper chromatography, we have the mobile and the stationary phase. Here also we are going to see the two phases, that is the mobile phase and stationary phase. Stationary phase, it is your matrix, and mobile phase, it is your ligand which you are going to use, because mobile phase, it is going to move towards your protein of interest or enzyme of interest. So specificity or the moving of ligand or enzyme towards its particular substrate, that is the mobile phase, and stationary phase, it is your matrix which you have used in that column. It may be a agarose or it may be any type of matrix. A stationary phase, it is consist of support medium on which substrate or ligand is bound covalently, and in which that are reactive groups that are essential for binding the target molecule, they are exposed. Your ligand can be attached to this spacer arm. Spacer arm is they are in the column which you are going to use, and in that spacer arm a ligand is attached. For that attachment of ligand covalently to the spacer arm, so that the protein of interest or the enzyme of interest can come and attach to the ligand, and you when there is the attachment or the binding, covalent binding of the ligand and the enzyme, the remaining solution, it is eluted from the column, and the protein of interest, by changing the buffer or the ionic strength of that particular matrix, you can purify your enzyme of interest. As the crude mixture of the substance is passed through the column chromatography, substances with the binding site from the immobilized substrate bind to the stationary phase, while the other substances, they are eluted. Okay, they are eluted from the column. And to take off the binding which is bind already to the ligand, we are going to change the ionic strength, or we are going to use the buffer, so that the detachment of the enzyme towards the ligand would be happen, and you can purify the protein or enzymes of your interest. Once the other substances, they are eluted, the bound target molecules, they are eluted by the methods including the complement ligand, that is in mobile phase, or changing the pH, ionic strength, or the polarity conditions. So here in this diagram you can see the methods. In first column you are going to see the target protein, the square, the rectangle, and the circle. They are the other components which is present in the solution. Okay, and the angles, acute angle, they are representing a ligand, and the yellow molecules, they are the affinity resins. Using a matrix or affinity resins, they are used here. Okay, and the red circles, they are the solute. In first column you are going to see a solution in which a target protein is going towards a ligand, and it is binding towards a ligand. Okay, then what we are doing, the affinity wash. Affinity wash means the molecules which have already binded towards a resin, they are in the resin, and the remaining molecules, they are taking off from the particular solution, that is we call them as a elute. Okay, the molecules, they are washed out. Then your protein, target protein or the enzyme which is already bound towards a ligand, they are going to be purified. For that what we are doing, we are changing the pH, or we are using a buffer, or we are using the ionic strength or the salt molecules to deattach that particular protein or the enzymes. By deattaching them, we can purify them. Okay, and these resins we can be used again in that particular chromatography. In the next diagram also you can see ligand are used, okay, and the analyte means a mixture. Okay, now what is happening over here? The specific ligand is used for a choice of the protein. Okay, so your protein is going to bind towards the ligand, the remaining is eluted from the analyte sample is eluted, and what you have now, you have only a ligand and your protein of interest. Now you have to wash out and you have to purify your protein of interest. Now you are using various techniques, that is the ionic strength or the changing the pH or using the ionic buffers. You are deattaching the selected protein or enzymes from the ligand and you are going to purify that. So now we have seen about the structure, how it is performed. We have seen its principle. Now we are moving towards the components. First, it is the matrix, an affinity column or affinity chromatography. In that column, you have a matrix in which a ligand is put, which is directly or indirectly coupled with your protein of interest. The matrix, it is the inert support to which ligand can directly or indirectly couple. In order to a matrix, it should be have efficient certain characteristics, which are matrix should be physical and chemically inert. It should be insoluble in solvent, because after your attachment you are going to use the buffer or ionic strength or changing the pH in which your matrix would be solubilized, and if it is solubilized you can't purify your enzyme. So your matrix should be insoluble in the solvents and buffers employed in the process. It should be chemically and mechanically stable. It must be easily coupled to a ligand or the spacer arm onto which ligand can be attached. It must exhibit good flow properties and have relatively large surface area for the attachment. The most useful matrix material, they are the agarose or the polyacrylamide. Now we are going to see about the second part, that is the spacer arm. First we are going to have seen about the matrix. Second, it is the spacer arm. Spacer arm, it is used to improve the binding property between a ligand and a target molecule by overcoming any effects of steric inheritance. We can see in the figure also here. You have a loading, you have the spacer arm in which the loading is going on, and after the loading only the molecules of interest or specific molecules, they are binding towards the ligand, and after the binding there is the deattachment of the molecules by using the various buffers and the ionic strength, and the protein of interest is eluted. Third, it is the ligand. A ligand, it is the molecule which have the reversibly capability to bind a specific target protein molecule. The ligand can be selected only after the nature of the macromolecules to be isolated is known. If you know the target protein very well or target enzyme very well, if you know the biological or the physical chemical properties of that particular enzyme, then only you can choose a ligand, specific ligand for that in this process, and it can be proceed. When a hormone receptor protein is going to purify by using this chromatography, the hormone itself is an ideal candidate for a ligand. For antibody, you can use an antigen, a hapten as a ligand. For enzymes, you can use an analog, inhibitor, cofactor, or the immobilized ligand for the purification method. Now the working. For the working, you have to first prepare the column in which you have to put a solid support, that is a saccharose or agarose or a cellulose material. Then you have to put a ligand which is selected according to desired isolate. Spacer arm which is attached between a ligand and the solid support. See in the figure also you can see the protein affinity. Again the repeated figure is repeated, that is the binding, the covalent binding, then the washing, and then the deattachment. Second one is the loading of samples. Solution containing a mixture of substance. First we have prepared a column which contains a solid material, that is a cellulose or agarose. Then we are putting a ligand in that. Then we are using a spacer arm, which is the solid matrix between a ligand and a gel. Then we are loading our sample. Solution containing a mixture of substances is poured in the elution column and allowed to run in a controlled rate. And then your third step is elution. When there is a binding of a ligand to a protein of interest or the target enzyme, then you have to elute the molecule. Target substances, they are recovered by changing the conditions, okay, to favor elution of the bound molecules. So this was about the affinity chromatography. This is how the affinity chromatography work. The three main things you have to remember in the affinity chromatography, that is a matrix, a ligand and a spacer arm, okay. And by using this matrix or by using these molecules, you can purify your enzymes of interest by using this technique. Its applications: the affinity chromatography, it is the most powerful method used for the separation and purification of specific product. It is essential a sample purification technique used for the purification of the biological product, that is the proteins or enzymes. It has the major application which includes the separation of mixture from the compound, removal of impurities in the purification technique. It is used in enzyme assay. For that you should know the enzyme activity. It is used in the detection of the substrates. It is used for the binding sites of the enzymes, using investigation which is actually a binding, where is the actual binding site of the enzymes on that particular enzyme. It is also used in vitro antigen antibody reactions. It is used for detection of single nucleotide polymorphism in mutation of the nucleic acid. What are the advantages? It have the high specificity, because it is a very specific method, so it is having the higher specificity. Target molecules can be obtained at the higher rate and in pure state. It is very important. Your target molecule you are going to get in pure state. It is single step purification method. The matrix can be reused rapidly. The matrix, it is solid and can be easily washed and dried. Give purified product in with the higher yield, and specific remove of specific components such as the proteases or other enzymes can be also done by using this type of chromatography. Now, what are the limitations or disadvantages of this type of chromatography? It is time consuming method. It requires some time for the purification. More amounts of solvents they are used, which may be expensive, that is your ligands. Intense labor. Non-specific absorption can be totally eliminated and it only can be minimized. It is high costly because your immobilized ligand, they are very they are highly costly. Proteins gets denatured if the required pH is not adjusted. If you fail to adjust the pH of the column, then a protein can be denatured or the enzyme activity can be denatured, and because of that the purified enzyme would be in non-active state. From the summary, what we have learned in this session, we have seen the protein purification method, which is based on the specific binding property, that is the affinity chromatography. We have seen its principle, its working, its component, its applications, its advantages and the disadvantages. Now in our next session, we have seen about the specific binding method. In our next session, we are going to see a method which is based on the charge and the molecular size, which is the ion exchange chromatography, which is based on the charge, electrophoresis, which is based on the charge, and on the size, that is the density gradient method or the gel filtration method. We are going to see its principle, its working, its components, its applications and advantages or disadvantages of that particular method. If you have any questions, you may ask. Thank you.